in human reproduction?
Yes, sir. So by the way, did you check the papers?
Yes, I did. You
Please
will get,
proceed.
yes, you will get the paper and the marks both probably after the class. I'll send it. I've checked all the batches finally.
So how was the score?
You will get the paper with remarks. Do you want me to give your remarks right here? No, I guess. Okay.
If it's good, so you can see.
Yes, the good thing about your paper is, Aisha, that you are improving from last year, and you are improving in terms of you are better understanding the questions. But I find, because I always want my students, if they are like A, then I wish for them to get A plus. If they're A plus, I wish for them to get A double plus. So it's like that. So you're doing good from where we started last year, where in orienting the answers, you are having less problems now, but still you need practice to orient your subjective answers, because see, subjective answers and oral answers are just like where to begin and where to end. That should be very, very clear in your head, right?
Yes,
So
sir.
sometimes you write things which just lengthen the answer, but do not give any value to that answer. It's like it's not asked. Even if you don't write that, it's okay. And sometimes you miss some important concepts and just end the answer prematurely. So I've given you all the remarks there in your answer sheet.
Okay.
Everyone okay? And then you'll get it. Take a look and complete the paper. Attend the complete paper, leaving anything.
Actually, I remember that there was a question about what we are saying cystogamy, and so I was cystogamy. I was knowing the answer, but like I wrote it at the end, and I just wrote some few words. I thought that if there would be more time, I would have elaborated it.
First thing is, you do not have to get uh, anxious or nervous if you do not know something. Second thing is, the kind of classes we engage in, when you hear some word in biology, you can understand something about it. Just have that confidence in yourself that when I will sit in the boards or in my school examination, this is for both of you, Nora and Aisha. Nora, are you hearing me? Am I audible?
Yes.
Yes. So this is for both of you. When you sit in the exam, you should sit with the confidence that I must be knowing something about this. If even if it is not the complete answer, it may be something around that answer. So if you leave any question in your school examinations in subjective tests, the empty space means sure short zero, right? There cannot be a negative marking in subjective exams.
Yes,
So, but if you leave a question, it is a sure short zero. But if you attempt it, if you try to write and explain some logic and tell about something, either it can be wrong or it can be slightly right or partially right. So if it is wrong, then it is as good as leaving the answer. But you tried, right? And what if some part of it is right and you get some partial marks? So never leave. Okay, attempt the full question. That's the key in biology subjective exams. Okay, Aisha and Nora.
yes,
Yes.
Okay, so let's start with from where we ended. I think we did some basic concepts and then we started with the male reproductive system. So quickly, just tell me what we discussed for the male reproductive system. Let's go to Nora. Nora, who's Nafia? Is she your sisters?
Yes.
Okay. Okay, Nora, let's start with you. Give me a quick recap of whatever it's. We did not discuss a lot about male system, but what did we discuss quickly from your notes and what is from the board in 5 minutes? There, Nora?
Yes.
Give a quick recap. Are you finding it difficult to where to start from?
Yes.
Let's start from here, the flow chart. So there is a sperm and an ovum. And what are these two? What is the
These
sperm?
two are gametes.
Yes, perfect. So let's start with gametes. And then what happens? Keep going.
Then fertilization.
Yes. And fertilization results in formation of
Zygote.
zygote. Yes. Now zygote is a diploid unicellular, like single cell.
Yes.
Then the zygote starts developing to become
An
embryo, which is multicellular.
multicellular.
Then the developing embryo is called
Fetus.
fetus. And this is all till before birth. Fetus till birth. After birth, it is called a neonatal baby, right?
Yes.
And then when it is 6 months, 7 months, some months old, it's called an infant, a toddler, and a child. And then it grows and develops to become comes adolescence. You know adolescence? Where
Yes.
puberty is reached, right? And then
Yes.
from there you become
Adult.
yes. And adults are sexually mature and they can again start forming gametes in their body, right? So you understand? And the same cycle continues in the adult body. Okay, Nora?
Yes.
And about the male system, we started with the organs, and I told you that the primary sexual organ is testes in a male. So let's start writing things about male system right now. Testes are the primary sexual organs, also known as male gonads. Testes are primary sexual organs, also known as male gonads. Male gonad is testes and female gonad is ovaries, okay? Now, testis is present in scrotum, and we also discussed why it is present in a bag-like structure outside the abdominal wall in scrotum. Because, did you write this in the last class?
Yes.
Because it requires a lower temperature than the body temperature to do spermatogenesis. And what is spermatogenesis?
Formation of sperms.
Yes, formation of sperms. Right. And we also, the last thing that we discussed in the last class was that testes actually are not formed in the scrotal sac. When they are formed, when they are developed in the abdominal cavity of the baby, right? As an embryo. And then it descends back, it descends down into the scrotum through inguinal canal, right?
Yes.
Yeah. So this you have to remember. Now let's come to this diagram and let's start understanding it. So let's say this is a testis and you see these testicular lobules, right? So write down inside the testis. Let me make you write things about testes. Write down the heading testes. Inside the testes, inside each testes, inside each testis, if it is a singular, it is TES, TIS. And if it is plural, it is TES, TES. One is testis. And inside each testis, there are around 250 testicular lobules. Okay? Clear? Both of you?
Yes.
These lobules basically are like chambers. So they are small compartments. Can write down. Testicular lobules are small compartments and inside each compartment, write down each lobule contains 1 to 3 seminiferous tubule. So it's like a compartment. So it's a small chamber, and inside that small room, there are 1, 2, or 3. Like it could be either 1 or 2 or 3 tubes. So 3 very small tubes called seminiferous tubules are present in each lobule. Is it clear? So if there are 250 lobules, how many total seminiferous tubules can be present in one testis? Maximum? Around 700, right? 700, 750 maximum.
Yes.
Yes, you understand? 1 to 3 in each, and let's say there is 3 in each, then 3 into 250 becomes 750. So somewhere around 500 to 700 seminiferous tubules are there in each testis, correct? And write down seminiferous tubule. Seminiferous tubule is the site of spermatogenesis. What do we mean by site of spermatogenesis? This is where spermatogenesis happens, correct? You understand, Aisha?
Yes, sir.
Yes.
So NCERT has shown testicular lobules. Inside it, there will be seminiferous tubules, which they have not shown. If you zoom it up and see, you will see inside each chamber. Yes,
Can I ask one?
yes, please.
So if the diagram will it come for labeling or for drawing?
Yes.
Or like
For labeling, it can come even in entrances, but to draw, if they ask you to draw, I have mostly seen that the question give you a choice that draw a well labeled diagram of either male reproductive system or female, and then you can choose female one. I always advise all my students to choose the female system because that's more straightforward and easier to remember. Okay, but that's why we are spending time here. That's why I'm explaining you this diagram so that you know which thing is there. So you understood testicular lobules are in the innermost part of the testis, Aisha?
yes, sir.
And inside it there are seminiferous tubules. Now this is where sperms are formed. So when they are formed, after that they start moving out of the testis. So when they start moving out, there is a network. So I'm using a red ink. Can you see here the red part which I have marked here?
Yes, sir.
So this region is called rete testis. Okay. The second part is called rete testis. So after testicular lobules, when you go out towards the outside, you will find rete testis. Now write down, rete testis is a network of tubules, you can say. So it's like from every chamber things are collected and they are brought in this one common region called rete testis. So it has networks coming from everywhere. Then it collects all the newly formed sperms and sends it to vasa efferentia, this third part, which you see here, right? Labeled. So vasa efferentia comes out of the testis. Rete testis is still inside the testis. It is collecting and then giving it to vasa efferentia, which is outside the testis. And write down, vasa efferentia is highly coiled tubules. Vasa efferentia are highly coiled tubules. They are very coiled. You can see here, right? And then vasa efferentia goes and let me use a blue color here and joins this one organ here. I think some other color, bright color will be better. Use green one. So do you see, can you see the structure that I'm marking with green? All of you can see, both of you can see this green marking of mine.
Yes.
this is a comma shaped, it looks like a comma or a crescent shape. So this structure is called epididymis. So vasa efferentia goes into epididymis. Write down epididymis, the very important organ for epididymis is a very important organ for maturation of functional maturation of sperms. Write down functional maturation of sperms. Okay. Then from epididymis is clear.
Yes.
For functional maturation of sperm. Then from there, when the sperms are mature, they go into this tube, which you see here as vas deferens. So they come out in vas deferens, where they are stored for some time. And then finally from there, they go into the ejaculatory duct and the urethra. Okay. So if we mark it in an order, so the first thing will be seminiferous tubule or testicular lobules first. Second, retitestis, then they go to vas. This is how sperms move out of the testis. Vas efferentia, then epididymis, then finally to vas deferens. All the ducts are clear to you? Then it meets, you see here, if I go to the above diagram, see this urinary bladder, everyone? Both of you? The urinary bladder and the ejaculatory duct. Do you see this duct also? These two ducts are joining at this region. Is this region visible to you? Where I am putting this first, this one, where this yellow part is marked. So at here, at this junction, from there onwards, there is a common passage for both sperms and urine. Because from urinary bladder, urine will come, and from this vas deferens, sperms will come. And they both join, and urethra is the common passage, this part. So they join somewhere here, and this part, urethra, you can write down, it's a common passage. Urethra is a common passage for urine and sperms. Okay. Clear? Any doubt in the diagram?
No.
No, right. Then now let's talk about these three glands. So you also see that there are three glands associated. I'm going to mark them with red. First is the seminal vesicle, the first gland. These are not ducts, these are glands which start secretion. Second is prostate gland. And the third is bulbourethral gland in series. So these three glands are just lined up after vas deferens and before urethra. Is it clear? And together, these glands secrete the semen, the fluid in which sperms are transferred. So is the whole diagram clear to you? Any doubt in the diagram?
No, sir.
Just look at the diagram and ask me if you have any doubt, then I'll move forward.
Actually, I want to ask you anything.
Yes.
The problem is with the drawing.
Sorry?
Actually, I want to ask you one thing, and that thing is about the drawing. So like,
How to draw it?
when we are drawing it, so can we draw it in the simple way? Like, it's so hard
Yes.
and confusing.
You just start simply. Start with, if it comes as a drawing, just practice.
Especially this epididymis and this vas efferentia, this part is so difficult to draw.
I think you just need to practice this twice and then you will understand. So you need to make a testes, make lobules in it, and then a retitestis, vasa. If you just know the sequence, you can just keep making it. It does not have to be like very, very aesthetically beautiful, but till the time the concepts are all there, then gland should be in line, and then finally the urethra, it's okay. Okay.
Yes, sir. Thank you.
Okay, let's start with the male system then. The system is clear to you, right? Now write down a question that we will start with. Write down a question. Explain the hormone, the regulation, bracket में write down hormonal. Explain the regulation of male reproductive system. Good evening, Nida. Am I audible and visible to you, Nida?
Yes.
So we just started, we discussed the structures, and we are just starting with the regulation of male reproductive system. Okay. So the question is, this is a question, write down this question. Let's talk about it. So how is a male system regulated? Now it is clear that male system, both male and female reproductive system are present right from the birth. Yes, they are present in the body right from the beginning, right? When we are born, these organs are there, right? Ovary is there, testes is there, yes or no? People, are you there?
Yes, sir.
So these organs are there, but they are not functioning, correct? They are not functioning because the body is not sexually mature till now. Then how does this regulation happens? Now I'll take you to class 11th, the chemical control and coordination part. Remember chemical control and coordination?
Yes.
And there you studied about hypothalamus. Remember studying about hypothalamus?
Yes.
So the hormonal control is like this.
It means that the brain will control the hormones, right?
Yes. So inside the brain, there is a structure called hypothalamus. Okay. And hypothalamus secretes a hormone known as GnRH. Anyone remembers the name of this hormone, GnRH? It's called gonadotropin.
So I remember it. I think so.
Yeah, it's called gonadotropin releasing hormone. I'm just telling you. GnRH, gonadotropin releasing hormone. Okay. Yes. Okay. Aisha.
Yes, sir.
And then this hormone goes and acts on pituitary gland. Remember pituitary gland, the master gland?
Yes.
Yes, sir.
Pituitary gland secretes a lot of hormones. Two hormones which pituitary gland secretes are under the effect of GnRH is LH and FSH. So it secretes LH. Remember LH? Luteinizing hormone. So it secretes luteinizing hormone. And the second hormone it secretes is called FSH. Anyone remembers the full form of FSH?
Follicle secreting hormone.
Follicle stimulating, not secreting, okay? Follicle stimulating, good. Hormone cannot secrete another thing, right? Hormone can stimulate something, right?
Yes.
So follicle stimulating hormone. Perfect. Now both these two hormones go and act on testes. In males, they go and act on testes. Okay, where in testes, write down FSH is important for or necessary for initiating spermatogenesis. So do you understand why spermatogenesis only starts at puberty? Because at puberty, this FSH hormone starts secreting from the pituitary gland. Is it clear? Yes?
Yes.
Yes, sir.
It goes and acts on the testes and it initiates the spermatogenesis process. Whereas the other hormone, which is LH, what does LH do? Write down. It stimulates.
Luteinizing hormone.
Yes, it is luteinizing hormone. The name is luteinizing hormone. What it does, the function is luteinizing.
What we are saying, I know, sir. That LH hormone, it controls the menstrual cycle, right? For females.
In females, yes.
Yeah.
But in males also FSH and LH is there. This
In males, what we are saying, in males, I guess, it makes testosterone.
yes, perfect. It stimulates the synthesis or secretion of testosterone from, there are special cells called Leydig cells. From Leydig cells of testis. Write this down. This is the function of these two hormones. Write this down. And what is testosterone? What is testosterone? Any idea about testosterone? Yes, it is the male, it's the sexual hormone of the males produced from the gonads. Now this testosterone, when it starts getting synthesized from the testes, the Leydig cells of the testes, it has two functions. The first function is testosterone is responsible for secondary sexual characters to develop. This is what puberty is, right? When secondary sexual characters develop in puberty, we also call it puberty is reached. Understand, everyone? This is the first function. And the second function of testosterone is to maintain spermatogenesis. Remember, spermatogenesis was started by FSH, okay, but is maintained by testosterone. Okay, everyone?
Yes.
Make this flowchart. This is the hormonal regulation of the male reproductive system. Is it clear? Yes, it's clear. Nora.
Yes, yes.
Yes, okay. So finish it and let me know, then I'll move forward. When you're done noting this down, we'll move forward.
Done, sir.
Others, Nora and Nida.
Yes.
Now, coming to secondary sexual characters, which is puberty. So let's talk about, write down a question here. What changes takes place during puberty in males? Tell me. Let's start with this. What are the changes which are taking place in males
High pitch voice.
because of testosterone? His first thing is facial hairs, right, Aisha?
Yes, sir.
Facial hairs include beard and mustache, right?
Yeah.
And then you were saying something about the voice. Tell me.
Yeah. High pitch voice.
High pitch. Sure, males have a high pitch voice.
Deep.
Males have a deep voice or
Yeah.
a heavy voice, right? Which is low pitched. A heavy voice is low pitched. A deep voice is low pitched. A high pitched voice is very shrill. Understand? Like females have high pitched voice.
Sorry, sir.
Okay, you understand, Aisha?
Yes, sir.
Females have a high pitched voice. Males have a low pitched voice. Now, why does this change in voice happens in males? Because of what?
Voice works that larynx.
Yes, voice box or larynx, which is also known as Adam's apple. Remember?
Yeah.
Adam's apple is visible. It is the protrusion. You can write down. Protrusion of larynx in the neck. Okay, you can see that in the neck, the larynx protrudes out after it develops. Protrusion of larynx, which is also known as the voice box, and because of which the voice becomes
Deep.
yes, coarse voice, or we also use the word deep voice. Heavy, deep, coarse. Basically, all of this means low pitched voice. Just remember that the pitch is low. Okay.
Then acne and pimples.
Sorry.
Acne and pimples.
Yes, that is yes. That is when hormonal changes starts happening. It also depends on the level of hormone in the body and how the body is dealing with it.
But
So it's lot of sebaceous glands they develop. They start secreting lots and lots of oil, and skin becomes oily. If you see a baby's skin is very soft, right? Like
yeah.
cotton candy, soft and dry. They do not profusely like start secreting oil. They do not get acne.
Yes, sir.
After a person reaches puberty, there are lots of oil secretions from the sebaceous gland, acne and pimples that starts.
They can try something with that oil. It's this much oily discount. Not only for boys, for girls also.
Yeah, it depends on skin type. You know, all boys or all girls do not go through the same amount of sebaceous secretion or pimples or acne. You know, it depends. But yes, that's true. Development of oil glands. And oil glands are known as sebaceous glands. Then what else happens?
Enlargement of scrotum and testes.
Yes, enlargement like development of scrotum and testes. Right. Enlargement of scrotum. Right. Testes. Because spermatogenesis is going to start now. And apart from that, other secondary sexual characters. Muscle mass development, right?
Yeah.
Males tend to develop muscle mass in the effect of testosterone. So muscle mass development and body hair, right? Apart from facial hair, body hair or pubic hair. Pubic hair are the hair in the in the region around the genitalia, the external genitalia and armpits. They are called pubic hair. They are related to reproductive maturity. The hair on our head, on the scalp, they have nothing to do with the sexual maturity, right? Everyone has hair.
Their height also. It will grow like the sudden growth.
Yes, so sudden shoot up in height. Height shoot up. Perfect. And
Only this much, no?
yes, mostly this much. Okay, cool. So just remember this. This is all the changes that happens in the males. We'll talk about changes in females also. But before that, let's also talk about one more concept called castration. You know about castration?
No.
No?
Yes.
No one knows about castration? You have a cat, right, Ash?
No.
But it's a female. Okay. So
I have male also.
you have a male cat?
Yeah.
How old is he?
2 years.
Okay, good then. So did you went to any veterinary, any doctor, or
Yeah.
did they suggested you to get rid of the male cat's testes?
Oh yeah. Yeah.
We do it with our pets, we do it with the domestic animals, males.
But why?
Removal of testes. We remove their testes. We cut the scrotal sac and we remove the testes. Or what we do is we just cut the spermatic cord, if not the whole testis. Like we just do not want, like basically we cut the whole testis. Like for animals, we don't care. We cut the whole testis and remove. So castration is right down. Removal. Then we'll come to Y part. Removal of testes surgically. But why are we doing it?
So they won't produce babies.
But why we don't want them to produce babies?
There will be lots of kids.
But a single male cannot produce babies alone, right?
Yeah.
So if you have one single pet, suppose let's say I have a dog who's a male dog, and that's the only pet in my house. And wherever he'll go with me. So it's not like he will sneak peek in the midnight, go and reproduce with someone and come back home with lots of babies. That's not the issue, right? What is the issue? What do you think? What happens?
Partner.
Sorry?
Like we need to bring a female to the home.
No. I guess probably that's interesting. So you're saying that probably because you will get a female also home, so you just want to get male rid of the testes. But that's cruel. But okay, it is cruel. But one more major reason is this. So in the effect of testosterone, animals, including humans, undergo a sexual drive, which is also known as arousal. sexual arousal. So they have a behavior where they want to mate after reaching sexual maturity. They want to find a female partner and mate. And that sexual drive can make them very, very, what we say, arrogant. You know, they're not docile anymore. And they also get frustrated. If
It
they,
happened like that in my home.
sorry?
With my, he's still so silent and cute, man.
Maybe the change is not that much. So they start scratching a lot when they are under sexual drive. Males, they become more arrogant. They become more frustrated. They scratch a lot, and they sometimes don't eat.
My cat was living here is having.
Yes, so they probably sometimes don't eat. So veterinary doctors always advise that, you know, get your pet castrated, removal of testes surgically. And write down, castration is done to domesticated animals and pets. Done to domesticated animals and pets to keep them docile. Keep them docile. Docile means submissive, like not very arrogant, not causing harm. Otherwise, if an animal is in sexual drive, it may show very arrogant phenotypes or behavior. Sometimes dog which are not properly castrated, when they reach sexual drive, they start barking and biting a lot. So they will just get angry with anything and they will just go and bite a family member.
Are those police dogs?
Sorry, come again.
Guys, the police dogs are there, right? They're so scary as they bark a lot.
Ah, yes, yes. No, but you know, even those dogs are also very docile and they are trained. They bark because they are trained to find something and bark, right? It may not be always related to the sexual drive. Okay, but it becomes easier to train these dogs if they are castrated, because the only thing that they will learn now is what we will teach them. They will not get into their own natural instincts, the wild instincts of sexual drive, and do whatever they want to do. Okay, now let's start with the organs of the male. So first, so there are, write down, there are two types of organs in the male reproductive system. There are two types of organs in the male reproductive system. The primary organ, which is only testes, and the secondary organ or accessory organ. So primary organ, write down, primary organ is testes, which produces sperms, which produces male gamete and male sex hormones. So it produces male gametes and male sex hormones. So this organ is the most important organ for the reproduction process of males. Other organs, they do not play a direct role in the production of gametes or hormones, but they help the system. They aid in the process of reproduction. So secondary or accessory organs, in this, the most important one is the penis as an organ. So let's first talk about penis as an organ. Write down. It is important for the transfer of gametes in bracket write sperms. Transfer of gametes from the male system into the female system. Is it clear? Is it clear, everyone?
Yes.
Now it has it is divided into three parts. The first is called the root or the base of the penis. So this is where it is attached to the body wall. Write down root or base of the penis, where it is attached to the body wall. Not very important, like for studying. But what is important is the body, or which is known as the shaft. So shaft is also known as the penile body. And this part contains, write down, it contains erectile tissues. This is the interesting part of the biology of this system. Erectile tissues known as corpus spongiosum. This is the name of the tissue which is present in the penis, which makes the penile structure erect during the sexual reproduction. Okay, corpus spongiosum, and also another one is corpus cavernosum. These are the two terms. Okay, these are that is what you have to remember. It can be asked in the entrance and not in boards. But you understand that the shaft or the body part has erectile tissues, and the name of these erectile tissues are corpus spongiosum and corpus cavernosum. Now, how do they work? Write down. As the word tells you, spongiosum. Have you seen the structure of a sponge, everyone? Aisha, Nora, Nida? Structure of a sperm. How does a sperm look like? Sorry, structure of a sponge. How does a sponge look like?
I know this, but I don't know how to describe it.
It has. Yes, Nora, please continue.
Full of holes.
Yes, it has space, right? And what happens? That space is capable of containing a lot of liquid, right? A fluid. Yes. You understand? So these tissues are also like sponge, and they have spaces where blood gets filled. So write down, during erection, blood gets filled in these tissues to make it hard and erect. This is the biology. So they work through blood vascular system. So there are lots and lots of blood vessels that are supplying to the penile region, and during the arousal state or sexual drive state, which happens under the effect of testosterone, lots of blood comes to these tissues and gets filled, okay, in corpus spongiosum. Is it clear, everyone? Understand?
Yes,
Yeah.
Yes. The last part is called the glans. The root, the shaft, and the glans. The glans is also known as the head part of the penis. Head or, yeah, head. The glans or the head of the penis. So root, shaft, and glans. And about the head, write down. It is covered by a loose skin layer called foreskin. Foreskin. This is important to remember because the removal of the foreskin is known as circumcision. The removal of foreskin is called circumcision. Okay, so this is a process which is carried out in many cultures where the foreskin is removed. It is surgically removed. So you might want to add the word the surgical removal. Write down the surgical removal is known as circumcision. And one more thing about this part, the glans, write down. The glans region contains many sensitive nerve endings. Okay, they are sensory. They are basically sensory nerves. So the glans part is very sensitive part. Okay, is it clear, everyone, about the male system? Let me know when you're noted, when you're done. Just note it. I'll be back in 2 minutes. Okay, I just need to get water quickly. Okay, Aisha, I'll just be back.
Yes, sir.
Okay, am I audible and visible? Done, everyone? Aisha, Nida, and Nora?
Yes, sir.
Yes, any doubts? Now let's come to the primary, write down. Now we are in the primary organ, which is testes, okay? Now, testes, write down, it is known as a dual organ. You know why it is known as a dual organ? You have studied this in junior classes. It's a dual organ because it does two things. It produces gametes through spermatogenesis. So write down, why is testis known as dual organ? So it acts as a organ and produces gametes, spermatogenesis. And second, it also acts as a endocrine gland and produce hormone. Which hormone does it produce? We just studied.
Testosterone.
Yes. Testosterone. Okay. So it has dual function, both to produce sperms and to produce hormone. Now in males, there are certain things about spermatogenesis that you need to know. So let's, we do not have to define spermatogenesis, right? It is the formation of sperms. And spermatogenesis in males, when does it start? When does it begin?
At puberty.
Yes. And when does gametogenesis begins in females?
Before, at the time of embryo development.
Yes, right. Even before birth. Before the female is actually born, she has primary oocytes in her ovaries. Tiny, tiny little ovaries with which she is born. She already has oocytes in those little organs. But males do not do spermatogenesis before puberty. So spermatogenesis begins at puberty. And when does puberty reach in males?
Around 15 to 14, 14 to, yeah.
13, yes, 13 to 15 years. Can you see? In males. And when does it occurs in females, Nora?
Yes.
When does puberty reaches in females? In males, it is 13
Around
to 15.
11 years.
Yes, around 11 to 13 years. So in females, it is clearly a couple of years early, yes, right?
Yes.
Yeah. Okay, now let's talk about the process of spermatogenesis. Can everyone see this figure here? Is it visible? Is the figure visible for spermatogenesis, everyone?
Yes.
Okay. Now, let's pay attention to this process and we'll understand this today. So this is a zoomed version of testes, okay? You can see there are compartment-like, small compartment-like structures here. So there will be small compartment-like structures, which will be called testicular lobules. Now, please keep telling me and let's see how much you remember from the above diagram. And inside the testicular lobules, what are present? Seminiferous tubule, right?
Yes.
Yes. And this is one tubule, one seminiferous tubule that they have taken out of the testes and shown as a zoomed in image. Can you see this?
Yes.
So in this, if you see this tube, in the center, so I'm using the red ink here. And see towards the center, what I'm drawing here, this is the empty space. Can you see this? Empty space?
Yes.
And this empty space is known as lumen, right? And from the lumen towards the outer wall, there is a thick, so this tube is very thick. It has a thick layer, right?
Yes.
In this thick layer of seminiferous tubule, so they have zoomed in this much. This rectangle is shown here, zoomed in. So if you see towards this part, this is the outer layer, which is this one. You understand, everyone, what is the diagram about?
Yes.
And towards here, this space that you see, this white space, this empty space, this space is lumen, which is this space. Okay, clear? So towards this side is the outer layer, and towards this side is the lumen. Now, spermatogenesis happens in this layer of seminiferous tubule. It does not happen in the lumen. Lumen is the empty space, so it happens in this layer. Now, in this layer, as we go down, okay, as we go down like this towards the lumen, sperm gets mature. Understand? So as we go down, maturation of sperm maturation takes place. Is it clear? So towards the top, you see this layer, everyone? But towards the boundary. Is this layer visible, everyone? Yeah. So towards the outer layer, let's first come to this chart, this flow chart. In the beginning, when testes are not yet formed and no spermatogenesis is happening, like in the embryonic stage, during the embryonic stage, there is first the primordial germ cells come up. Okay, so it all begins from primordial germ cells. This happens during the embryonic stage. This is formed. Okay, finally this gives rise to spermatogonium. Okay, so from this line, the dotted line that I'm making here onwards, this part of the flow chart is happening in testes. Okay, this was happening during embryonic stage. Primordial germ cell was formed when testes were not formed yet. These cells give rise to both the gonad and the gametes. You understand, everyone? Yes? Is it clear? Primordial germ cells are present during embryonic stage, and when testes is produced as an organ, when it is formed in the embryo, and when the baby is born, and when the baby reaches puberty. Okay, so after reaching puberty, the primordial germ cell divides mitotically. So there is mitotic division, means mitosis is happening. You know what is mitosis and meiosis, everyone? Everyone is clear with the concept of mitosis and meiosis? Yes or no? Nida, Aisha?
Sir, can you repeat again?
Do you want me to repeat the question or mitosis and meiosis?
Not the question. What you was asking?
The question is, both of you are familiar with mitosis and meiosis?
Yeah.
As mode of cell division? Nida?
Yes.
Yes. So you know that in mitosis, it's called equational division. There is no reduction of the genes, right? Of the chromosome number.
Yeah.
So in the beginning, primordial germ cells divide through mitosis and gives rise to spermatogonium, these cells. So these cells which you are seeing here are spermatogonium cells here, marked here. Okay. And the spermatogonium cells are present. So these are the spermatogonium cells. They are present towards the outermost layer of the seminiferous tubule. Can you see they are towards the very top, at the very end, somewhere here, right, of the tube? Is it clear? As the stages progress, so now the spermatogonium, this is the first stage in the testes, then the spermatogonium will again divide through mitosis. Okay, so here also mitosis is happening. Okay, so what is the ploidy of primordial cells? Let's also talk about the ploidy. So this is diploid. Okay, everyone, primordial germ cells are 2n. Clear?
Yes.
Then what will be the ploidy of spermatogonium cells? The division here is happening by mitosis. So will the ploidy change?
No.
No. So it will be 2n here also, right?
Yes.
Then spermatogonium forms primary spermatocyte. These cells here. What will be the, now again mitosis is happening. So what will be the ploidy of primary spermatocyte? Nora? Nida?
Will it be N?
No. So that is the catch here. This is a question which confuses a lot. So write down. It's a neat question. The term spermatocyte looks like it is a sperm cell, right? Because technically the name says that. Yes or no, Nora? Spermatocyte. Yes or no? So sperm cell. So you will think it is haploid, but no, it is diploid. Primary spermatocyte is formed from spermatogonium through mitotic division. So primary spermatocytes are diploid cells. Write down. This is a neat question. Make a star here. Primary spermatocytes are diploid cells. Written, everyone?
Yes, sir.
Now, and also write down, primary spermatocyte is the first cell line. Primary spermatocyte is the first cell line in the process of spermatogenesis. In the process of spermatogenesis that undergoes meiosis. Okay.
Sir, can you repeat?
Primary spermatocytes are the first cell line in the process of spermatogenesis that undergo meiotic division, meiosis, that undergo meiotic division. Okay.
Yes.
So primary spermatocyte starts the first meiotic division, M1. You know, meiosis has two internal cycles. One cycle of meiosis has two cell division cycles, meiosis 1 and meiosis 2. So from meiosis 1, one cell divides into 2, and with meiosis 2, those two cells further divide into 4. And when the cycle ends from one cell that we started with, we get 4 haploid daughter cells in meiosis, right? This was the process, remember?
Yes.
Yeah. So write down, primary spermatocytes undergo meiotic division 1. Primary spermatocytes undergo meiotic division 1 to form to form secondary spermatocyte. To form secondary spermatocyte. Now tell me, what will be the ploidy of secondary spermatocyte?
N.
Yes, this will be haploid because meiosis has started. So meiosis has two cycles, meiosis 1 and meiosis 2. And meiosis 1 is reductional and meiosis 2 is like mitosis. It's equational. Okay. Is it clear? So in meiosis 1, 2N, so this was a primary spermatocyte, but it was 2N. So this 2N becomes N and N. Okay. Do you understand? So the ploidy has reduced, right?
Yes.
And after meiotic division 2, so the secondary spermatocyte undergo meiotic division 2 to form. So this N will make N, N, and this N will also make N and N. So there will be 4 cells, all N. So you see in meiotic division 2, the N is not reducing further. It is staying the same, right? But in meiotic division 1, 2N reduced to N. Yes? Is it clear, everyone? Is it clear or no?
Yes.
Aisha, Nida? Nida?
Yes.
Yes. So write down, secondary spermatocyte is a haploid cell. It's the first haploid cell. Secondary spermatocyte is the first haploid cell in the process of spermatogenesis. Okay. This was asked, make a star. So you have to know which is haploid and which is diploid in spermatogenesis. These are neat questions. Now write down, secondary spermatocyte undergoes meiotic division 2. Secondary spermatocyte undergoes meiotic division 2, giving rise to 4 haploid spermatids. Giving rise to 4 haploid spermatids. And what are spermatids? What is the difference between a sperm and a spermatid? Anyone?
Sperm, it's how to say. So I'm saying. Alright. Sorry, I'm saying.
Yes, yes. Yes, please.
Um. Um. Uh. Uh.
So
So let me say.
each A for each sperm. There are 4 sperms for A. What do you think? Look at the figure here, Aisha. This will help you. So this is spermatid, this one, these 4, and these are mature sperm. So what is the difference between a sperm and a spermatid that you see?
Sperms are matured.
They're structurally mature, right? You're correct. You're not wrong. Just because I'm smiling and smirking does not mean you're wrong. You're correct. So every time you start answering, it just reminds me of how we started last year.
So please, it was really fun.
Okay. So write down. Spermatids are structurally immature. Structurally immature. and do not have a head and a tail like structure, and do not have a head and a tail like structure. Spermatids are also not capable of fertilizing. They cannot fertilize an ovum. So write down, spermatids are non-viable. They cannot fertilize a ovum. Okay. Is it clear? Spermatids cannot fertilize an ovum.
Yes.
So this process of formation of mature sperm from spermatids then undergoes where they get their head tail like arrangement. But before coming to this, let's come till spermatid. Let's see where they are in this layer. So we saw that spermatogonium, the first cell, are at the very top, right? Look here, everyone. Then where is primary spermatocyte? Below it, right? It has come down. This is a primary spermatocyte, right? Do you think that it has come slightly down towards the lumen?
Yes.
Then secondary is this one. They have marked these are secondary. They have come even more down, right?
Yes.
And then spermatids are these cells. These are spermatids which are developing. So they have come even more down. So you see from top to bottom, as they are progressing, they are coming down towards the lumen. Yes? Yes or no?
Yes.
Because ultimately, after they are mature and they become sperm, they are released into the lumen. They are released here. And then they move through the lumen and they come into this network. Can you see this network here clearly, which was not visible in the last diagram? Now tell me, what is this known as? This is the rete testis. Okay, everyone, are you following?
Yes.
So this is the network like structure called rete testis. And can you see these tubules, convoluted these tubes? Everyone can see this noodles like thing. Yes or no?
Yes.
They are vasa efferentia. I think it's getting clear now, right? And then this is epididymis clearly. And this is vas deferens. Is it clear, everyone? So write down spermatids undergo structural development where round spermatids undergo a series of changes to become streamlined What do we mean by streamlined? Streamlined, it is the shape that helps in movement in water or in air, right? Birds are streamlined and fish are also streamlined when they are moving, correct?
Okay, I was going to see.
Yes, right. So streamlined, motile,
It helps to move without any lubrication, right?
yes, without friction. Perfect, Aisha. Streamlined and motile spermatozoa. So spermatids become spermatozoa, and this process is known as from here to here spermiogenesis. It is called spermiogenesis. So if they ask you define spermiogenesis, this is the question. So write down the question, define spermiogenesis. We just answered that question. Spermiogenesis is clear, Nida? Nida? Is spermiogenesis clear?
Yes.
Yes. So it is a process of developing of the spermatids, which are round and non-motile, into spermatozoa, which are streamlined and motile. Okay. Now, after this maturation happens, after structural maturation happens through spermiogenesis. Now, let's come to one more thing. Do you see here Sertoli cells? Anyone knows what are Sertoli cells? See, these are these yellow cells which are present. Can you see this? I'm making a boundary of one Sertoli cell. Can you see they are big cells? This is a Sertoli cell. Then there is another Sertoli cell. Can you see these big cells, yellow cells? Everyone, you will see that
Yes.
every stage of the spermatogenesis is embedded, like is attached with a Sertoli cell, right? Yes. So Sertoli cells, write down. Sertoli cells. These are randomly scattered big cells in the seminiferous tubules. These are randomly scattered big cells in the seminiferous tubules.
Say?
Yes, Aisha. Welcome, Nora. Am I audible to you, Nora?
No, I was here only.
No, Nora. Nora
Acha.
just left, I think, in between.
Say,
Nora, am I audible to you?
Yes. Yes.
Yeah. Yes, Aisha, you have a question?
No, actually, I want to ask that today also we will have that extra question stuff.
Sorry, what?
Like the previous class we were discussing, so today also we will have, or we will just finish
The
this
questions. Yes, we will do that. So, is this clear? So, Nora, we were discussing Sertoli cells. You know what are Sertoli cells? Have you heard of these cells, Sertoli cells here? I have drawn the boundary
Yes.
of some. So they are big, write down, they are randomly scattered big cells in the seminiferous tubules that provide, that support and provide nutrition, that support and provide nutrition support and provide nutrition to developing spermatozoa. to developing spermatozoa. Okay, to the sperm cells. Is it clear?
Yes.
Also, write down. They also transport the sperms towards the lumen, towards the lumen of seminiferous tubule. Now remember, I was talking about that, see they are shifting down, right? But you did not ask me that when they are spermatogonium and primary oocyte, secondary, sorry, spermatogonium, primary spermatocyte, secondary spermatocyte, and spermatids, till this stage they cannot move, right? Then how are they moving down? So these Sertoli cells are pushing them towards the lumen slowly. Is it clear?
Yes.
And ultimately, when they reach at the very end, when they become spermatids, the spermatids put their head into the Sertoli cells to develop properly in the last phase, which is known as Permeagenesis. And when Permeagenesis is done, then the mature sperm is released in the lumen. Okay. Mature sperm is released in the lumen by the Sertoli cells. Write down. Mature sperms are released in the lumen by the Sertoli cells, and this specific process of release is called spermiation. So it's a very important question. Make a star on these three things. Spermatogenesis. What is the difference between spermatogenesis, spermiogenesis, and spermiation? Is it clear? The three things? Is it clear? Yes or no?
Yes, sir.
Can you repeat?
Yes. Question. The question, you want me to repeat the question? It's an important question that has come. What is the difference between spermatogenesis, spermiogenesis, and spermiation? Spermatogenesis, you all know, right? The overall process of formation of sperms. Spermiogenesis is the development of spermatids, which are round and non-motile, into streamlined motile spermatozoa, the last phase. And then when it is done, spermiation is the release of those developed sperms into the lumen. Okay? Clear, everyone? Everyone is written, yes or no? Please say it.
Yes, sir.
Perfect. Sertoli cells is also clear to you?
Yeah.
They support their, and also write down one more thing. Sertoli cells are known as the nurse cells. Why are they called nurse cells? Because they are like nurses. They take care of the spermatozoa. You know, this is what nurses do. Take care of the patient, move the patient here and there when the patient is not able to move, take it to the place like to washroom or somewhere, and then bring back, change the clothing, give the medicines, right? Energy, nutrition. Yes or no?
Yes.
Sertoli cells do the same, all the things. Now, please quickly make this, at least this flow chart, and label everything. And if you are, if you can take a screenshot of this. I'm just putting this right here. Yes. So I give all of you 2-3 minutes to finish this, and then we'll start with some neat questions. Okay. Okay, everyone. Those who are noting, please note down
Yes.
the flow chart and let me know when you are done. In the meanwhile, let me questions. You all can still see the screen, right? Am I?
Yes, sir.
You can still see the spermatogenesis thing, or are you seeing the?
You can see that the same thing spermatogenesis.
Okay, good.
Screen.
Yes, right. I wanted to see the screen. So I'm finding the previous year questions for this topic that we are discussing. So I have shortlisted some topics. So we finished spermatogenesis. Okay, this is a very main concept and a major. This can come as a 3 marker, 4 marker, or a 5 marker question also in boards. And lots of questions come in the entrance, have come in previous years from spermatogenesis process. Everything about the arrangement of the testes, the ducts, and the process of spermatogenesis is clear to you, all of you?
Yes.
Let me know when you're done, everyone. Noting down the, at least the flow chart, everyone should note if you are not able to note down the diagram.
Done, sir.
Done?
Yeah.
Just one moment. Let me. Here. Yes. Did we discuss about human reproduction? Questions from human reproduction in the last class also?
No.
No? Okay, let's do it then. For that, I need to stop sharing the screen. Can I stop sharing the screen?
Yes.
Yes, sir.
Yes. So you can see me, right? Aisha and Nora?
Yes, sir.
No.
You cannot see me? Am I not visible?
No, you're
I can see you.
Okay. Now I am sharing the questions. Okay. Previous year's questions. Okay. Can you see on your screen previous year questions from human reproduction?
Yes, sir.
Yeah. So I would first, like, this came in 2016. Second question. Aisha, please go with the second question and tell me what do you think is the right answer.
Which of the following depicts the correct pathway of transport of sperms? Testes, efferent ductus, epididymis, ossifrans, testes, efferent ductus, ossifrans. Testes, ossifrans, efferent ductus, epididymis, efferent ductus, testes, ossifrans, epididymis.
Do you know, have you understood the correct pathway? Like, it was the first thing that I started with. Remember 1, 2, 3, 4, 5 I marked, and it is the
Yeah.
question that came in NEET. So you have to, sometimes they ask you very simple questions, but if you have not paid attention to the diagrams, you will not be able to score, because here there is no logic that you can use. If you know it, you know it. If you do not know it, you do not know it. So retetestis, yes, tell me which one is the right answer, Aisha, according to you.
Testes, efferent ductus, epididymis. A.
A.
Yeah.
Okay. Nora.
Just a minute.
Yes. And in entrances, you will not get this much time. Okay. So try to be faster. And when I'm asking, I'm just going alternate. So next question will be for Nora, but Aisha should also solve it in that meanwhile. And again, yes, please write the answers to me in chat. Let's see how fast can you do it. So I'm also opening the chat here. Can you? Okay, you cannot see it. So Aisha has registered her answer.
Yes.
Nora, please register your answer in the chat to me, just to me, not to everyone. Okay. So after both of you have given the answer, I will just discuss the right one. And you both will just assess why you were right and why you were wrong. Okay?
Okay.
Nora and Aisha?
Yes. I'm having that feeling.
Okay. So for this, both the answers I have received, and the right answer is A.
I said correct answer.
Okay. Now let's see why it is A. It is A because retrotestis is the network inside the testes only, right? Nora, you understand? So retrotestis has to be the first because it is inside the testes. Before retrotestis, what is it? Seminiferous tubules, where the sperms are produced. So from seminiferous tubules, they go into retrotestis. Then from retrotestis, they go into vasa efferentia, which is called efferent duct also. So efferentia word or efferent word is same. Did you get confused by this, Aisha or Nora?
No.
No. Okay. So retrotestis, vasa efferentia or efferent duct, then epididymis, which efferent duct basically joins the retrotestis with epididymis, and then finally vas deferens. Is it clear, Nora?
Yes.
Yes, you understood?
Yes.
So if you understood, then now please, for you, the question is question number 7. Okay. But you have not studied we have not studied female system, but let's go to question number 7 anyways. I will tell you. Nora, you will read question 7 and give me the answer in the chat. Aisha, time starts now. Please read. Nora, you have to read also.
Select the incorrect statement. LH and FSH decrease gradually during the follicular phase. LH triggers secretion of androgens from the Leydig cells. FSH stimulates the Sertoli cells which help in the spermiogenesis. LH triggers ovulation in ovary.
So I just wanted to, can you attempt this? Aisha is attempting.
I know the answer.
So the question is asking you incorrect statement, not the correct one. Okay. So always pay attention. Sometimes the question comes, which of the following is not a reptile, for example. So you have to focus on the not word. If you just do not read the question properly, chances are you might end up choosing the wrong option. Nora, any idea what could be the right option here? A, yes. So both of you are correct. It is A because LH triggers secretion of androgen of Leydig cells that I have already told you. And FSH helps in spermatogenesis. So LH triggers ovulation in ovary. So I'll come to this question again when I will teach you female system, because I think then you will be able to answer it from your side. Right now you're just understanding it. Okay. Let's see any question which has come from male system. Yes, there is one which I shortlisted. Yes, let's come to 2015. Question number 13. Aisha, will you read this? This is what we have taught from the part that we have learned today. Please read
Which
question.
one of the following during gametogenesis is normally diagnosed for
What
mitochondrial?
answers quickly in the chat. Both Nora and Aisha, quickly.
I answered.
I have not got your answer, Aisha.
Not. Did you got it now?
Is it the same problem that's happening that happened last time also?
I think
I
so.
think
I'm typing, it's not coming.
yes. I got
Okay,
5
fine.
together. Okay, yes, the right answer is spermatogonia, right? We just studied spermatogonia, and which other cell is diploid after spermatogonia? What it becomes?
Primary spermatocytes.
That is also diploid, right?
Yeah.
Yes.
Primary spermatocyte. Sorry.
Yes, perfect. So you understand the questions now.
Yeah.
So this is very common one. The shared terminal duct. Look at question 17, Nora. I think you will read it and tell me the answer quickly.
The shared terminal duct of the reproductive and urinary system in the human female is urethra, ureter, vas deferens, vasa efferentia.
Yes, human male. What do you think is the answer? Just tell me. It's simple.
Urethra.
Yes, it is called urethra. Okay. Vasa efferentia is, which is, you know, everything. Ureter is the duct from kidneys to urinary bladder. Vas deferens is from testes to outside. Vasa efferentia is inside the, like joining the testes and the epididymis. So urethra is the common duct. Okay.
Yes.
Most of the questions are from female parts. So when we'll come to it.
So, by the way,
Yes.
female reproductive part carries more marks.
It does not carry more marks. It is more complex. So if you want to make difficult questions, it's highly likely that difficult questions can be made from female part. When we'll come to the female part in the next class, you will get to know why. Okay. Not as straightforward. Male system is straightforward, right? Question number 26, quickly. Aisha, please read, and Nora and Aisha have to write the answer in the chat.
One of the following statement is false in respect of viability of mammalian sperm.
It's asking false. Which one is false?
So you got the answer.
I received Nora's. I received two answers from Nora.
The second one.
Which one should I consider, Nora?
Second one.
Okay.
Sir, can I say the answer?
Yes, please.
It's yeah, 1 minute. So let me read it again. It's a sperm is viable for only up to 24 hours.
Okay, is this false? Is A false?
Wait 1 minute.
B, C, and D true.
Yeah.
Yes, the right answer is A because that is the false one. It is asking which one is false. So both of you have given the right answer, which is A. Sperm, write down what is wrong in this.
It can last for several days.
It can last for 2 to 3 days, 48 to 72 hours.
Yeah, till 48 hours maximum.
Yes. In some cases 72 as well, but 48. Let's write down. It can stay up to 2 days, 48 hours. Both of you write down. Sperms can be viable for 48 hours. Are you also noting down the questions? No. Just solve.
I'm just taking the photo.
Okay. So you understand that survival of sperm depends on pH. That's true. Why? So Nora, you understand why C is not the false one? Because viability, it depends on motility. Viability is the ability to fertilize the ovum. The ability of the sperm to fertilize the ovum is called viability. Is it clear? And if a sperm is not motile, it will also not be able to fertilize. Correct?
Yes.
Yes. Now, question number 32. Nora, please read question number 32.
If for some reason the vasa efferentia in the human reproductive system gets blocked, the gametes will not be transported from testes to epididymis, epididymis to vas deferens, ovary to uterus, vagina to uterus.
Yes. So if you know that vasa efferentia is a part of male system, these two, C and D, are already gone, right? They cannot be the answer. So which one will be the answer? Aisha? You are taking so much time for this. This is the
No,